Let's begin by talking about the endomembrane system. This refers to a network of membranes that exist within the cytoplasm, as well as a few other organelles that are bound by membrane. So the endomembrane system is responsible for the production as well as processing and digestion of things like lipids and proteins, as well as other large molecules. The system is also going to be responsible for recycling and disposing of molecules and particles that we no longer use. So the key components of the endomembrane system includes the following. There is the nuclear envelope that we talked about, and we know that the nuclear envelope is going to be continuous with the endoplasmic reticulum, a huge network that exists in the cytoplasm. And there are two types of endoplasmic reticulum, rough or smooth. There's something called the Golgi bodies or Golgi apparatus, as well as another organelle called the lysosome. So let's take a closer look at the different components of the endomembrane system and their functions. So the first part we're going to talk about is the rough endoplasmic reticulum, or the RER. The word rough in the name actually refers to the roughness that the endoplasmic reticulum appears to have when you view them under high power microscope, and that roughness is actually due to the presence of these ribosomes on the reticulum. Okay, so again, endoplasmic means within your cytoplasm, and reticulum basically means a network. Okay, so you can see there are two types of ER here. The rough ER has ribosomes on them, and the smooth ERs they do not. In fact, the rough ER, as we mentioned in the previous video, is a continuous membrane with the nuclear envelope. It's physically connected to the nuclear envelope. The ribosomes on the rough ER, they are going to be synthesizing proteins directly into the rough ER. You can see there's a ribosome here, and it's going to be producing the proteins by decoding the messenger RNA. We talked about the process of translation briefly, where you are going to use the instruction in your genetic code to create proteins. And these proteins are then going to move to the edge of your ER, where it will bud off and be surrounded in a membrane brown vesicle, and this vesicle is going to move to another part of the cell. Now, typically there are two types of proteins that the rough ER is going to be producing. You can be producing membrane proteins, and these membrane proteins are actually going to be traveling to the surface of the cell, where they will embed themselves in the plasma membrane. Another type of proteins that the ribosome can produce would be what's called the secretory proteins. So these are proteins that are destined to be released from the cell, and they either types of proteins are going to be traveling in a special transport vesicle, and they are going to move to the appropriate place, either in the membrane, or they're going to be secreted. And actually, in some cases, you might not be secreting the proteins immediately. You might be storing it in your cytoplasm for later use. So cells that secrete a lot of proteins, such as cells of your salivary glands, these are the glands that gives you saliva, they actually have a lot of rough ER in them to keep up with the demand of the secretion, because in saliva there are actually some enzymes that helps you break down sugar, and those enzymes are actually proteins. So in order to and you keep on making a lot of saliva like 24/7, right? So in order to keep up with the demand of those enzymes in the saliva, the salivary cells are going to have quite a lot of rough ER. Next, we have the smooth ER, and you can see under the high power microscope here that the smooth ER does not have those black dots that you see in the rough ER. Okay, so no ribosomes for the smooth ER, and there are ribosomes on the rough ER. There are many functions associated with the smooth ER. One of them is for the synthesis of various lipids, including steroid hormones, for example. So you would expect to find a high number of smooth ER in cells of the ovaries and testes, where they will be producing the sex hormones. So testosterone in male and progesterone and estrogen in female. They are also important for breaking down fat as well as detoxifying drugs. So one of the organs in the body that helps detoxify drugs would be your liver. And so if liver cells are exposed to a drug, then the amount of smooth ER and its detoxifying enzyme actually increases over time. And this can strengthen the body's tolerance for the drug, meaning that a higher dose will be required in the future in order to have the same desired effect. Okay, so you kind of increase your tolerance because now you are able to metabolize these drugs much faster. So in some cases, if you are using antibiotics in a very chronic way, okay, and you use it even though it's unwarranted for, maybe due to wrong prescription, then what happens is they might decrease the effectiveness over time because your liver is able to break them down much quicker. And in fact, increasing tolerance to drugs is one of the hallmarks of addiction, something, of course, that is potentially going to have serious consequences if you are going to increase the dose of certain drugs, such as opioids, for example. So there, that is the function of the smooth ER. Next, we have something called the Golgi body or the Golgi apparatus. So it's called the Golgi body because it's named after the Italian scientist Camillo Golgi, who has basically discovered the organelles. And the Golgi body is a layer, consists of several layers of membrane stacked together. And the function of the Golgi body is basically to receive the vesicles that are produced by the rough ER or vesicles that are going to be ingested by the cells. So earlier, just let me skip back a few slides to show you. We talk about how the ribosomes would be producing the proteins directly into the ER, and that's going to be budding off in a vesicle. So these vesicles might be ready to go to its final destination right away, but oftentimes it's not quite ready yet, and so those vesicles are going to be arriving at the Golgi body. And the Golgi body is going to basically sort the content of those vesicles. They are going to modify it in some cases, repackage it, and tag them so that they end up at the right place. Okay, so in some cases, the repackaged vesicles are going to be stored for later use, and in some other cases, they are going to be targeted for immediate release. So basically, the Golgi body, you can think of them as like the mail sorting center. Okay, it kind of tells things where to go and when to go. So one of those vesicles that are coming off from the Golgi actually would form another important organelle, and that organelle is called the lysosome. Lysosome contains various digestive enzymes in them, and these enzymes can be used for breaking down macromolecules. These macromolecules might be things that we have ingested, the cells that might have taken up macromolecules from your bloodstream. You might break them down in the lysosome, and here is a picture of what that looks like. This is your cell. There's some food particle. The cell can sometimes ingest those food particle and surround them in another structure, like a food vacuole, we call that. That will then fuse with the lysosome, where the enzymes in the lysosome will break down the food and digest it. So now we can use that products that we break down from the food, and we can use it to build our own molecules, for example. Another function of lysozyme is to recycle old organelles. So here is a damaged organelle; it's no longer functioning, and so we don't want to use up resources if it's not working. So the lysozyme is going to fuse with that damaged organelle. They're going to recycle it, and we can kind of recover some of the materials from the digested organelles, and we can use it for other purposes within the cell. Sometimes lysosomes are going to release all of its enzymes into the cytoplasm of the cell, and in that case, the cell is going to die as a result of all these digestive chemicals being released simultaneously. And that type of behavior is sometimes deliberate. For example, during embryonic development, we need the cells of the webbing between our fingers to kind of die off, so that our fingers are going to not be connected to each other. And those kind of timed programmed cell death often involves the lysosome. So for that reason, lysosomes are sometimes called the suicide sac, because it will cause the cell to basically self-destruct. There are a group of diseases that are collectively known as the lysosomal storage disease. These are typically inherited disease where one or more of the lysosomal digestive enzymes are going to be missing. Remember, inside the lysosome is a mixture of these enzymes. Each of the enzymes are going to have a specific function. And so if you're missing some of these enzymes and they turn out to be involved in some key process, then it's going to cause quite a bit of problem. So a lot of these lysosomal storage diseases are actually fatal in early childhood because those enzymes are so crucial in the cells, and without them, the cells might not be able to function. So there are two examples of these of the lysosomal storage disease that I want to share with you. First one is the Pompey's disease. In this case, you don't have the enzymes to break down complex sugar in the body, and that results in an accumulation of harmful amount of glycogen in the muscle cell. And you know, this will result in the weakening of the muscle, particularly in the heart. And so if nothing is done, and I'm not sure if there's anything that can be done to fix these problems. I don't think there are cures for them at this point. And so if the muscle of the heart starts to weaken, then it will lead to early heart failure, which of course can be fatal. Another example is something called Tay-Sachs disease. In this case, the lipid digesting enzyme is going to be missing in the lysosome. And if you have an accumulation of these excess lipids in the nerve cells, then it will result in interference with brain development, and actually they're going to cause the neurons to die. And so of course, that is also going to be lethal. But fortunately, since these are genetic diseases, many of them can be screened even pre-conception. So parents can get genetic testing and genetic counseling to see if they carry a disease version of the gene that causes Tay-Sachs disease, and based on that information, they can decide whether or not they will have a family. So that is why lysosome is quite important. Without the enzymes in the lysosomes, there are often going to be very detrimental diseases. So just to quickly recap what we have been talking about in the endomembrane system. The arrows here shows some of the pathways of cell products distribution and membrane migration through the vesicle transport. So for example, we can have a digestive enzyme that is going to be created by the rough ER over here. So the ribosomes will make that digestive enzymes directly in the rough ER, where the finished proteins will then move to the edge of the rough ER, and it's going to be budding off in a transport vesicle. So that vesicle is then going to be traveling to the Golgi apparatus shown here, which is multiple layers of membranes, and that vesicle will merge with the Golgi to be processed. Okay, so during the processing period, there might be other things that are added to the proteins. There might be tags that are going to be added to the vesicle, so that the final product is going to know where to go. So over here we can see after sorting, the protein is repackaged in a new vesicle, and that new vesicle can actually travel all the way to the cell surface and release its content. Okay, so that might be true if you are producing a hormone, okay, and you need to release that into the bloodstream. In other cases, if we are talking about the digestive enzymes again, then it might get packaged into a new vesicle that will actually form the lysosome. Okay, and then the lysosome is going to hang around in the cytoplasm. And they will be breaking down worn out organelles. They will be digesting macromolecules, and it will continue to function that way until you need to make more lysosome. Then the whole pathway will just kind of repeat itself. Okay, so this is a good summary for you to kind of think about in terms of what's going on within the endomembrane system. So before we talk about other organelles, I think it's a good idea to maybe take a pause and go to our study guide, and we try to label as many things as we can from this diagram. So if you haven't already, please go ahead and download a copy of this study guide from eCentennial and come back to the video, and we're going to label it together. Okay, so here is an animal cell, and you can tell an animal cells apart from a plant cells because you can see in the plant cells there is going to be another structure outside of the cell membrane, which is this cell wall. We haven't really talked about that yet, but we will later on. And also in the plant cells, there are usually this big organelle inside to hold some water, and we don't see that in an animal cell. So let's see what we can identify in this picture. We talked about the cell membrane. Okay, so this thing here, this outer edge, that would be the cell membrane, and we can label that together, cell membrane or plasma membrane. Doesn't really matter which one you use. Okay, so the plasma membrane is made up of phospholipid bilayer primarily. There is a little bit of cholesterol in it to control the fluidity. And what's really important about the plasma membrane is that they are selectively permeable. They let some things in and some things out, but not everything. In the center of this cell, you will see the nucleus. Okay, so this whole thing is the nucleus, which is where you will find your genetic material. Not all cells are going to have the nucleus, right? Only the eukaryotic cells will have a nucleus. So animal cells have a nucleus, and over here we can see the plant cells also have a nucleus. And the nucleus is going to be surrounded by a membrane. So the membrane here is called the nuclear envelope. Okay, nuclear envelope. And that nuclear envelope is going to be continuous with the endoplasmic reticulum. But before that, you can see that there are also these nuclear pores on it, right? The nuclear pores allow things to go in and out of the nucleus. For example, if you make a copy of the DNA in the form of RNA in a process called transcription, then the RNA can exit through the nuclear pore into the cytoplasm. Another thing is there is something called the nucleolus in the center here. So nucleolus is where you will be making new ribosomes. So those ribosomes will also be coming out into the cytoplasm through these nuclear pores. Can't really see it too clearly here, so I'm just gonna cheat a little bit. I'm just gonna draw my own genetic material here. So these genetic materials are basically DNA that has been packaged with histone proteins so that they can all fit into your nucleus. So those packaged DNA, we call them chromatin. And they will condense further and become chromosomes just before cell division. What else did we learn so far? We learned about the endomembrane system. So the nuclear envelope is part of the endomembrane system. We have the rough ER here, rough endoplasmic reticulum, and you can tell that it's rough because of the dots that are on the surface. I can zoom in a little bit for you. You see all those blue dots. Those are the ribosomes that are covering the rough ER. The rough ER produces membrane proteins as well as secretory proteins that might be released from the cell. Over here we have the smooth ER. Right? The smooth ER is usually a little bit away from the nucleus, and they don't have the ribosomes on them. Okay, and so smooth ERs are responsible for lipid synthesis, like steroid hormones. They are going to help with the detoxifying process, as you will find in the liver cells, for example. And over here we have another one. Over here we have the Golgi body. Golgi body or Golgi apparatus. And the main function of the Golgi body is to be receiving vesicles that are released from the ER. So there is a vesicle that's budding off. There's another vesicle that's budding off, and then maybe another one over here. So all these vesicles are going to go to the Golgi body, where they're going to be modified, repackaged, and sorted. And some will be released from the cell. Others, they might form organelles that will stay in the cytoplasm. And one of those organelles that are part of the endomembrane system is going to be the lysosome. Okay, so there is a lysosome, and there's another lysosome, right? These lysosomes contain special enzymes that will help you break down macromolecules, break down food molecules. They could break down worn out organelles, right? And in some cases, they might even cause the cell to destroy themselves, and that's something that's important during embryonic development. So I think that's all we have looked at so far. Let's just quickly go to the plant cells and identify the same thing. Should be quite easy. You can try it yourself first. There is the nucleus. Since plant cells are eukaryotic cells, that's why they also have a nucleus. And I'm just going to skip the nuclear envelope and the nucleolus stuff. It's there is a nucleolus. Is very straightforward. You can definitely do that yourself. Over here we see the rough ER, and over here we see the smooth ER. And on the left here we have what's called the Golgi body. Golgi body. Okay. That's it for now. And I mean, you kind of see these things and it looks like the lysosome that we've labeled on the previous diagrams, but they are actually not. Plant cells actually do not have lysosomes, and they have an equivalent of that to perform similar functions, but we're not going to be going into details for that, so just kind of ignore these circle stuff. Let's go back to the PowerPoint slides. We will learn a few other organelles in the cell. The first one is peroxisome. Okay, peroxisomes are small round organelles that will be carrying out oxidation reactions. So basically, it means they will use oxygen in the reactions, and as a byproduct, they release something called H2O2. And the H2O2, which is called hydrogen peroxides, are actually harmful to the cells, and so they must break them down into water and oxygen before it harms the cell. So the peroxisomes are important for breaking down fatty acids and amino acids, and they will help detoxify many poisons that enters the body, such as alcohol in the liver. Okay, so you might feel like this is very similar to the function of lysozyme. It kind of is, right? There is a little bit of overlap, but it's important to know that the peroxisome is not part of the endomembrane system. And in fact, plant cells, they do have peroxisomes, but they do not have the lysosome. On a diagram, it's hard to tell. If this is just a black and white diagram, how do you know this is the peroxisome and how do you know that's the lysosome? You don't, okay? It's just a cartoon drawing, right? Like for it's anybody's guess, right? Which one is which. So I'm not gonna be asking you these kind of trick questions on the test. You just kind of have to know that the lysosomes function and it's part of the endomembrane system, and you have to know the peroxisome function as well, but you won't have to be labeling them on a diagram. This is an actual microscope slide, and you can see the lysosomes are a little bit darker here and the peroxisomes are a little bit lighter, but like I said, for the most part they are both circular organelles, and it's really difficult to distinguish them unless you have look at many slides, right? So don't worry about identifying them on the diagram. That's the bottom line. Next, we have something called the mitochondria. Okay, the mitochondria is the site of energy production. You remember we talked about cellular respiration earlier? That's where you take glucose and you will break it down to convert it into carbon dioxide and water, and in the process you're going to be creating energy, right, in the form of something called ATP. So that process, that cellular respiration process, takes place in the mitochondria. Okay, it's the powerhouse of the cell. So some cells only have a few mitochondria. Others, they would have more than 1,000 of them, right? Depending on the energy need of the cell. Right? If you are looking at a heart cell, heart muscle cell, which constantly beats throughout the lifetime, you would expect to have a lot of mitochondria. So the mitochondria has a double membrane surrounding it. There is the outer membrane, which is shown here, and then there is the inner membrane that is all folded up. Okay, perhaps it's better if I draw it for you. There is the outer membrane, okay, and then there is going to be the inner membrane that is going to be all folded up like so. So between the outer membrane and the inner membrane, this space here is called the intermembrane space. Okay, intermembrane space. It's important that we know the structure of the mitochondria, because in lecture 5 we will look at the events that are taking place in these different parts of the mitochondria in order for the body to produce ATP, to produce usable energy. On the inside of the inner membrane, there is going to be fluid in here, and that fluid is called the matrix. Okay, and finally, these foldings that are formed by the inner membrane, the finger-like projection, the finger-like foldings, they are called cristae. Cristae. Okay, so that's what a mitochondria looks like, and again, that's going to be the powerhouse of the cell. So let's just quickly switch to the diagram again. Just keep that next to you so you can flip back and forth. There is the mitochondria. Okay, plants, they also need to produce energy, so they would have mitochondria as well. mitochondria if it's singular, and if it's plural, then we say mitochondria. Okay, we change it to A instead of on. So over here, there is one, right, and then there is another one. Okay, so I'm pointing at two, so we will say mitochondria. Okay, singular is on, plural is A. Okay, we're not going to label the peroxisomes here. It's hard to tell where they are anyways, so we'll just leave that be. Okay, let's go back to the PowerPoint slides and see what is next. Next, we have chloroplasts. Chloroplasts are organelles that are found in plant cells and not animal cells. They contain something called chlorophyll, and chlorophyll is a pigment that basically makes the plants green, and they use the chlorophylls to carry out photosynthesis. The chlorophyll is the compound that is able to absorb sunlight and use that energy to combine with carbon dioxide to make oxygen and glucose for the plant. The chloroplast is also a double membrane organ, just like the mitochondria. So there is the outer membrane, there is the inner membrane, and inside the inner membrane, there are these fluid-filled membrane structures called the thylakoids. And in the membranes of the thylakoids, that's where you will be able to find the chlorophyll. And the fluid that is filling up the space between the thylakoids and the inner membrane, that's called the stroma. So the stroma is kind of the equivalent of the matrix in the mitochondria. So here in this picture, you can see all these kind of circles with the green fillings. Those are going to be your chloroplasts. And here is a single plant cell. Here is another single plant cells, and these are all the chloroplasts that you'll find in them. So the relationship between mitochondria and chloroplasts is actually quite important. We briefly mentioned how the mitochondria is where cellular respiration takes place. That's where you will be taking glucose and oxygen and to produce carbon dioxide, water, as well as energy. Okay, so then the carbon dioxide and the water are going to be released from the body. And for the chloroplasts in plants, is actually the opposite. Plants are able to use the carbon dioxide and the water, as well as capturing energy from the sunlight to then convert them into glucose and oxygen. Okay, so they store that glucose as starch. We learned that in lecture 2, and the plants can then use that in their own mitochondria. Okay, so both animals and plant cells, they have mitochondria. Okay, and plant cells, they also have chloroplasts. So the difference is this: the plants, they are able to use the chloroplasts to produce their own glucose, and then they will burn the glucose in their mitochondria for energy. Okay, in humans, instead of making our own glucose, we will be consuming the glucose from our food source. Okay, so you will be eating, say, a potato. That would be the starch that's created by the plants, and then you will use it to produce our own energy. So there's an interdependency between the function of the mitochondria and the chloroplast. Okay, and in the case of plants, these two things exist within the same cells, but for animals, we would have to be consuming the glucose from from the food source instead of producing it ourselves. Another thing that's found in plants are these big vacuoles, and big vacuoles often takes up as much as 70 to 90% of the volume in a plant cells, and they are mostly important for storing water as well as nutrients. But in some case, such as the cells of a flower pebble, the central vacuole might contain some pigments that gives the flowers its beautiful colors. And other cases, there might be poisons in these central vacuoles to protect the plant from being eaten by animals, for example. In some single-cell organisms, such as this paramecium again, they live in fresh pond water, and so there's a constant influx of water from the surrounding. Okay, there's a lot of water going into the cell all the time, and that could cause the cell to swell up and potentially burst. And so to prevent themselves from bursting, they have these specialized contractile vacuoles, which are basically pumps that will take the excess water out of the cell so that it doesn't burst. So that requires energy to do so, and that energy, of course, is going to be supplied by the mitochondria. Another thing that is found in plant cells but not in animal cells are these cell walls. Okay, so the cell wall exists outside of the cell membrane. We learned that in lecture 2, the cell wall is made up of the cellulose, which is a type of polysaccharide. They are very, very strong fibers, and that's actually going to protect the plant cells from bursting. Right? Plants, they need a lot of water, right, to survive. They need the water to perform photosynthesis, and so all these extra water will cause the cell to swell up, and again, to protect it from bursting, the cell wall is going to allow for some rigidity, as well as help the cell to kind of prop up the plants when they are filled with water. Okay, so the last part of the lecture, we are going to be talking about what's called the cytoskeleton. These are specialized fibers inside the cell that are going to help with maintaining cell shape. They're going to help things to move around in the cells, and there are three types of cytoskeleton that we're going to be looking at. They are called microfilaments, shown here with two braids of long circular fibers. There's something called the intermediate filaments, which comes in bundles of how many here? 1, 2, 3, 4, 5, 6, 7, 8, 9. And then we have these microtubules, which are kind of like hollow tubes. Okay, and each of them are going to have unique functions. Let's begin with microfilaments. Okay, microfilaments are made up of two intertwined actin protein strands. Okay, so these circle balls, they are a protein called actin. Right? In fact, we have a lot of actins in our muscle. Right? We mentioned this earlier in lecture 2. Muscle is made up of primarily two proteins, right? Actin and myosin. And so it turns out actins also make these microfilaments. Okay, so you have two long actin protein strands, and they are kind of braided together like that. You can find microfilaments throughout the entire cell. All these green fibers here are the actin filaments, and they actually provide attachment sites for various organelles. So without the actin filaments, the organelles would just be randomly sloshing back and forth in the cell, right? We don't really want that. Okay, so by having the actin filament there, it kind of gives them a scaffold to kind of hang on to, and so the movement is actually not a random process. Okay, the microfilament actually also allows cells to move. They can reorganize the actin structure and the location. For example, the cell can break down the actin here and start to build more on this side, and that will help the cell move in this particular direction. The next one is the intermediate filament. Intermediate filaments connects to structures that helps you lock the cell membranes together. So let's say you have a cell here in the small intestine, okay, and then there's another cell next to it. Between the two cell membranes, you will have these intermediate filaments that would help seal the gap between them and really lock them together. Okay, because if this is the intestine, you don't really want the content to kind of leak all over the place, right? So by creating these tight junctions, it prevents the cells to have gaps between them. Okay, and it kind of controls the movement of material that way. The nuclear envelope is actually supported by an inner layer of intermediate filaments called the lamins. Okay, and what happens is if the lamin is mutated, then it could affect the nuclear stability. Okay, and if the nucleus is not stable, then chromatin structures and the gene expression are going to be affected. So that mutation in the lamin gene is actually related to a condition called progeria. In progeria, you have basically accelerated aging in the person. So this is just an 11-year-old boy, but you can see he has all the feature of an old person, and that's caused by the mutation in the lamin A gene. In fact, there is a movie that was made about this condition. It was by Robin Williams. I don't know if you guys ever seen that. You can probably Google it and see a trailer of it or something. It's not an exact accurate portrayal of the condition, but nevertheless it has the same premise as the accelerated aging. Last but not least, we have these microtubules. They are small hollow tubes, and they provide long tracts for the vesicles to move around in the cell. So if you have a vesicle that butts off from the rough ER, it needs to kind of find its way to the Golgi body, right? And we don't want to rely on random movement and random diffusion for the vesicle to find the target. That might take too long, right? So there are actually tracts that are formed by the microtubules, and there are carrier proteins called kinesin that will actually walk along the microtubules while carrying the vesicle. Okay, so they will transport the vesicles to where they need to go. The microtubules are also excellent for structural support. In this picture, you can see the microtubule shown here in yellow is actually guiding the growth direction of a nerve ending, okay, so that you're going to be able to make the correct nerve connections, right? And so these are the functions of the microtubules. In animal cells, there are also going to be a pair of organelles called the centrioles. These centrioles are only found in animals and not plant cells. And what they do is they actually produce microtubules fibers called spindle fibers. Okay, these spindle fibers are only going to be created when the cell is dividing. And we will learn more about this process in lecture 7 or 8, I think. And what happens is these spindle fibers, these microtubules, are going to help you guide the movements of the chromosomes. Okay, remember the chromosomes are the condensed version of your DNA, right? And so you need to tell these DNA where to go in the cell division. You want to make sure that if one cell divides into two cells, you want to make sure that each of the cells are going to get the equal amount of genetic material. So to guide the chromosomes to go to the right place, that's what the spindle fibers are going to be for. Okay, so again, the centrioles are where the spindle fibers are created, and they are only found in animal cells but not plant cells. There are also these things called cilia. We talked about cilia briefly earlier in the lecture. They are found in your airway, for example. They will help remove trapped particles. You will also find cilia in the female reproductive tracts to guide the movement of the eggs. And these cilia are also made up of microtubules. Okay, so usually a cell would have multiple cilia, and they would just kind of beat back and forth to create motions and guide the movement of an egg, for example. And over here in sperm cells, we have a tail, and that tail is called the flagellum. Okay, so usually just one flagellum or two per cell. In humans, definitely just one. A sperm with two tails is actually not going to be able to go anywhere. It just kind of swims around in a circle. But in some other single-cell organisms, like the ones that live in the pond, there could be two flagellum per cell. Okay, and instead of beating back and forth, they kind of beat in a wave-like motion. Okay, to help propel the cell forward. And the flagellums are also going to be made up of the microtubules. Here is a quick summary of what's unique in an animal cells and what's unique in the plant cells. Okay, we have the lysosome in the animal cells, the centrioles, and the flagellum. But in plant cells, unique to them would be the chloroplast, central vacuole, and the cell wall. And that is it for the lecture. But before we go, let's go back to this diagram and see if there's anything else we missed that we should label. So up here we have the centriole. Centrioles. Remember, the centrioles are important for creating the spindle fibers, which are made up of microtubules, and they would guide the movements of chromosome during cell division. And I think that's all we can label for this. You can kind of see some cytoskeleton here, right? So these fibers, it's hard to tell which type they are because of the diagram. It's not very detailed drawing. But these lines in the background, that would be your cytoskeleton. Okay, just gotta draw an arrow and point to it. And it could be intermediate filaments. It could be the microtubules. Is kind of hard to tell from the diagram. In the plant cells, we have the chloroplast right here. I'm going to label that for you. That's the chloroplast, which contains the chlorophyll, and the plants are going to need the chloroplast to do photosynthesis. There you can see there is the central vacuole. It stores mostly water and nutrients, and in some cases they can store pigments or poison to protect the plant. Anything else we have here? in the plant cells, yep, there is the cell wall, and the cell wall is outside of the cell membrane. Hard to tell the cell membrane in this picture, but I'm just going to draw it in for you. Right? That would be the cell membrane that I'm coloring right now, and so the cell wall is going to be outside of the cell membrane. Okay, so just label that for you. The pink thing right here, cell membrane. Okay, so that's it for this lecture. Make sure you do the rest of the study guide, and we'll take it up at a later time. Thank you for watching.